Okay, so hello and good evening to one and all present here. I am Srijan Sen, and today I will be taking the week 11 live class session on the course Fundamentals of Protein Chemistry, which is being taught by Professor Shagata Das Gupta of IIT Kharagpur. So today's topic will basically be on protein interactions. Okay, so I'm sharing my slides with you all. Alright. So in the previous classes we talked about the proteins, their purification strategies, the different types of proteins and all, and then we talked a little bit about the DNA and how you can undergo recombinant DNA technology and all. Though it was not a part of that particular week, but yes, that gives you a better idea about the information on the DNA level as well, because after all, it becomes a very important factor in protein engineering. So today we will be going into the characterization, the important or the advancement of proteins, that is the protein-protein interactions. So we know that protein-protein interactions are universal to life, and they have roles like in different cell signaling networks. Then it has different regulatory functions: cellular regulation, DNA translation, transcription, and even in DNA replication, some cellular level work. Apart from that, uh, protein-protein interactions play a very important role in different cell signaling networks that are associated with the hallmarks of cancer. So therefore, protein-protein interactions becomes a very attractive topic of study. So it happens when two or more proteins bind together. Proteins control and mediate many of the biological activities of the cell. So mostly different any kinds of activities is controlled by a protein. Now there are several functions or biological activities that occur inside the cells that needs to be controlled by a particular protein, or a particular interaction of different proteins. Can be a protein-protein, can be protein ion interactions. Like we know that there are several molecules or ions that act as cofactors for the activity of the different proteins. So therefore it is important. So today mainly we will not talk about the protein ion or protein carbohydrate lipid interaction. It will mostly cover protein and another protein. So what we need to know about protein-protein interactions is that it improves the understanding of the diseases, and it acts as attractive target for new therapeutic approaches. The structure of protein influences its functions by determining the other molecules with which it can interact, and the consequences of these interactions. There are several multi-subunit proteins like the hemoglobin. Then you have the RNA polymerase, small ribonucleoproteins, and the ribosome that carries out its function in not as a monomer, but there are different subunits. Each of can be of its own type or a different type, and it carries out its function. So transient protein interactions, like strong are reversible. It undergoes changes in its oligomeric state. Now there are interactions of several protein kinases, protein phosphatases, glycosyl transferases, like you can see over here. Okay. Then you have acyl transferases, proteases that interact with the substrate proteins to carry out the different functions. Like several catalysts are present that play important role by interacting with its substrate proteins. So proteins that are important for the cellular growth, cell cycle, metabolic pathways, signal transduction, all carries out their activities through protein-protein interactions. Now, therefore, these interactions are very important. Although can be disease prone, these interactions can be disease prone, or it can lead to Alzheimer's, Creutzfeldt Jacobs disease and all. But also they are very important in some important signaling networks, like there are several SH3 domain interactions that are important for the down regulation of the epidermal growth factor receptors, the EGFRs, that are important for controlling of cancer and all. So it is useful, as well as it can lead to several diseases, so it has both the pros and cons. So therefore, the most known example of protein-protein interactions which we have studied about is the actin-myosin while regulating the muscular contraction in our body. It's an important protein-protein interaction. Now we'll get into the examples of a few protein-protein interactions, like there is a signal transduction, that is the activity of the cell regulated by the extracellular signals. You can see that there is it's a combination of you can see collagen present over here. The glutamic acid is highlighted, you have the midas, and then it leads to signal transduction through these alpha helices, and that are highlighted in red. So transport across the membranes is a very important phenomenon, where a protein carries another protein across the cellular membrane. Cell metabolism and muscle contraction. So cell metabolism happens in many biosynthetic process. Enzymes that interact with its substrates, or even with itself, to produce small compounds or other macromolecules, that's the product. Then muscle contraction, where the myosin filaments act as molecular motors, and by binding to this actin, it enables the filament sliding. Now, protein-protein interactions can be of different types. Like, it can be categorized on the basis of their composition. Like, you have the homooligomers, where the macromolecule complexes have only one type of protein subunits, like protein-protein interactions in the muscle contraction. Then you have the heterooligomers. Okay, so the heterooligomers, these are the macromolecule complexes of multiple protein subunits. Like have a protein A, and then you need protein B or a protein C to come and bind to its active site, or at a position. Okay, so it can be its active site, or it can be. So mostly what happens for enzyme is that the active site remains vacant. It binds to a site other than the active site, and it triggers it. Okay, so this triggering process is very important for the carrying out of the function, or for the enzyme to bind to its active site. Like, we had studied about the lock-in key model and the induced fit model. So like an enzyme of an induced carrying the activity of an induced fit model, what it does is there can be a protein A that is the enzyme, or an enzyme A, and you need a protein A to activate that particular enzyme, the active site of the enzyme, and only when the protein A comes and binds to the particular enzyme, the active site gets activated, and then it can bind to the substrate to carry out the function. So that's a heterooligomer. So it happens in cytochrome oxidase, TRPC3, that's the transient receptor potential cation channel. So there are some channel proteins, some that are in the form of heterooligomers. Now also you can categorize on the basis of their duration, that is stable interactions, they comprise of the interactions that last for like longer durations, so they have a very tight affinity towards each other, so the KD value is like in nanomolar or picomolar range. And so these interactions carry out the functional or the structural roles, like the hemoglobin structure. Okay, and then there are some transient interactions, where the interactions last for a shorter period of time. So these the different types of the durations of these interactions are very important or critical, because you do not want some interactions to be for a long duration. Like, even if you have a higher affinity, it should be for a purpose or a role. Like, there are some receptor kinase receptoring pathways that actually need the transient interactions more than stable interactions, because if there are some pathways that are upregulated or overexpression of these receptor pathways happen, then it can lead to certain kinds of diseases. Mainly cancer happening pathway is majorly due to this kinds of situations. But the interaction is very tight, and the proteins cannot dissociate, leading to an upregulation of the pathway and an overexpression of the cancerous or the proteins that can lead to cellular proliferation and cancer. Okay, so therefore it's important that this particular type of interaction is known, or the knowledge about that the protein or the protein-protein interaction that we are studying is basically known to us, and their roles and functions. So the effects of protein interactions can alter the kinetic properties of the individual proteins. That's absolutely true. Now, protein interactions are one common mechanism to allow for the substrate channeling. It can result in the formation of a new binding site, like I was mentioning for, say, for a particular enzyme. Even if the protein binds to the in the active protein comes and interacts with that particular enzyme in the binding site, then it might happen that another channel or another part of that particular enzyme might get activated, that can also carry out a similar kind of a function. So it can lead to the formation of new binding sites, and also the binding site can be very different from the active site for the enzyme. So we need to be very much sure, because the active site is an active site, because we know an active site of an enzyme to be the most active part of that particular enzyme. The amino acids work hand in hand to actually carry out the different functions. Now, these interactions, protein-protein interactions, can also inactivate a particular protein. That is also one of the drawbacks. Now, it can change the specificity of a protein for its substrate. That can be advantageous or disadvantageous depending on whether it's binding to the active site, then it acts as an inhibitor, but also it can be a protein as well. Also, if it's binding apart from the active site to a site that can activate the particular active site, then what happens is that the activity gets enhanced, and the protein, the enzyme is able to carry out its function more smoothly. So the catalytic efficiency, we know, we have studied about the catalytic efficiency of the enzyme, so the catalytic efficiency is increased. Also, the function of the different proteins, like there are different transcription factors that binds with the DNA, or it requires partner proteins to interact with this to bind to the DNA. So there can be a lot of activities that happens inside. So transcription factors are basically, it's proteins that interact with nucleic acids, different promoter sequences, and different DNA or RNA sequences. So it's important to identify the different interactions to understand the extent to which they take place in the cell, and determine the consequences of the particular interaction. So, like, shortly we spoke about the homooligomers. So in the basis of composition, we are segregating them. So homooligomers are macromolecular complexes that constitute by only one type of protein subunit. So protein oligomer complex assembly, the subunits is guided by the different non-covalent interactions in the quaternary structure, like the protein, in the interactions happening in the muscle contraction. Like there are several enzymes, carrier proteins, transcriptional regulatory factors, like I was mentioning, that carry out the functions as homooligomers. So the other part is the heterooligomers. They have distinct protein subunits that interact to carry out the function. So these are essential to control cellular functions like the cytochrome oxidase or the TRPC3. That's the potential cation ion channels, and these are very important. So for different functions, like you see the alpha and the beta subunit of the hemoglobin. So these are the heterooligomers. Now, if you segregate them on the basis of their bonding, it can be like covalent or non-covalent. So covalent is a strongest association or a stronger association, like there can be the disulfide bonds or the amide bond formation. So there is an electron sharing between the elements, and there is a bond formation like the disulfide bonds between the two cysteine molecules. Apart from that, there can be like post-translational modifications like ubiquitinations, sumoylation. So these things can happen, and that those things are like the covalent interaction or affinity for one protein towards the other. The non-covalent interactions are quite more simpler. So there is a transient interaction by the weaker bonds like the hydrogen bonds, ionic interactions. You have the van der Waals forces, the hydrophobic bonds, and also these are the different transient interactions that also play an active role. Like it's not important for a protein to always have a covalent interaction, like I was mentioning. It Even if there is a transient interaction, it can carry out its function. When the function is over, it just dissociates and allows the monomeric form of the protein to be in its inactive state or carry out the other functions that does not require the help of its partner protein. Now, the some post-translational modifications includes the ubiquitination, where you have the degradation of defective or superfluous proteins, like a single chain polypeptide. Now, ubiquitination is an enzymatic post-translational modification in which the ubiquitin is attached to a substrate protein. So the steps includes the activation of that particular protein, then you have the conjugation, and ligation happens for the post-translational modifications. So by ubiquitin activating enzymes, you have the E1, E2s, and the ubiquitin ligases like E3. So there are several ubiquitin ligases that are very important for the downregulation of different kinase pathways, receptor pathways, the cancer causing pathways like the MAPK. Then you have there are several other pathways like AI3KT. So there are several cellular pathways that can lead to cancer. So the ubiquitin ligases are very important proteins in the down regulation. So what they do, they interact with the proteins that were recruited by the kinase domain. So the kinase domain recruits certain proteins after they are activated by the extracellular ligands that comes and activates the receptor. So after the activation, the kinase recruits certain proteins. So these proteins are important for carrying out the downstream signaling, but this downstream signaling needs to be stopped or halted. So for that, you need the ubiquitin ligases, that is the ubiquitin ligase E3. Okay, so these are the some halters or the stopping enzymes for different cancer causing pathway. Now, like on the basis of interaction, we had studied like for lasting for a short duration of time is a transient interaction of a protein and a protein. So like you have the G protein coupled receptors that transiently binds to the GI or G0 proteins that are activated by the extracellular ligands. And there are certain stable interactions that interacts for a long time or a permanent complex forming as subunits. So it carries out like the functional or the structural roles. Example is the cytochrome C. So the cytochrome C and the CCO complex, so it's stabilized by a few electrostatic interactions. So there are involvement of salt bridges, electrostatic interactions between the long chains with small contact surface. So in contrast to the other cytochrome C complexes, there are numerous water molecules that are found in the long intermolecular span between cytochrome C and the CCO. So you can see there is a docking that is happening. So there is an electrostatic interaction, like the blue beads show that. So there is an electrostatic interaction that actually stabilizes the protein-protein interaction, and that is a type of a stable interaction. Now, so this is about the different types of interactions or the protein-protein interactions that can happen. Now this is a bit of information about the protein domains. Like interactions are possible only due to structural domains within the proteins. Like it cannot be an extracellular portion that is not attached to the protein or a dissociated portion. It has to be a part of the protein. So a protein domain is conserved part of a given protein sequence, and the tertiary structure that can evolve, function and exist independently of the rest of the protein chain. Now proteins hold structural domains that allow their interaction with and bind to specific sequences on other protein molecules. So you have the phosphotyrosine containing motifs, like the proteins that carry the motif have the activated receptors for the different growth factors, cytokines and the antigens. Like the recognition protein-protein motifs, interaction motifs includes the SH2 domains. So that's a phosphotyrosine containing motif. It's a very important motif and function. So SH2, SH3 domains are present in different proteins, and they are involved in different kinds of interactions with the proline-rich regions. So SH3 domains are known to interact with proline-rich regions, and so proteins containing these proline-rich regions, a stretch of prolines in a particular consensuous sequence, can bind with this particular domains. And also you have the PTB domains that binds to unphosphorylated peptides or proteins. Now you have the phosphoserine threonine motif. So these recognizing protein-protein interaction domains is the 1433 proteins, then FHA domains, then WW domains, or the uh, okay, so you have the WW domains, or even SH3 domains have this tryptophan motif. It's conserved in most of these SH3 domains, and this is responsible for binding to the proline rich region. And it also binds to unphosphorylated peptides and proline rich sequences. Now there are certain bromodomains and chromodomains. Okay, so acetylation of the lysine residues, so the proteins who carry the motif histones, so they are recognizing the proteins creating binding sites for the bromodomain. And the methylation of the lysine residues, these are also proteins that carry the histones, carry the motifs, so those are known as histones. And these recognizing proteins, these histones creates binding sites for the chromodomains. So methylation of lysine in the histones are responsible for binding sites of the chromodomains, and acetylation of lysine in the histones are creates the binding sites for the bromodomain to bind. You do not need to actually remember this, but it's a sort of an information. Like for apoptosis, there are uh, protein-protein interactions, that domains that are involved. Like for apoptosis, you have the DD domain, that's the death domain, the dead domain. You have the card BH14 Okay, for like dimerization, you have the SAM, that's sterile alpha motif. Vesicle traffic, you have the GYF, snare, VHS. Okay, for chromatin, you have the CSD, cold shock domain. So most of them you might not be aware of, but it's not very important for people to remember. Like you just need to know like there are certain protein-protein interactions that are important for like proteolysis, chromatin formation, apoptosis, dimerization, vesicle trafficking, and so on. Now, so SH2 domains are important. So they have different cell. They are involved in different cell signaling networks, or they are involved in different cellular functions that because for which we cannot actually ignore them. So these SH2 and SH3 domains are mostly important for different actin polymerizations, okay, different cellular communications, cellular signalings and all. So individually they have a role. Like the structure is mostly of a two alpha helix and beta strands, and it has high affinity for phosphorylated tyrosine residues. So it is known to identify a sequence of 3 to 6 amino acids within a peptide motif, and it represents a larger class of phosphorylated tyrosine recognition domains. So, like similarly, SH3 domains or the SRC homology 3 domains, they also are involved in like the actin polymerization and cellular communication. SH3 domains have you have heard about the ubiquitin ligase I was telling. So there are certain SH3 domains like the GRB2 SH3 domain and the beta pix SH3 domain. So the SH3 domains of these proteins, so these are big proteins, the beta pix and the GRB2. So these have different other motifs, the PHDH motif, the proline bridge sequences. Okay, so but the SH3 or the SH3 two domains are very critical. So these are the few important domains that you need to have information about uh, and have very important roles inside the cells. But what is more important is like how do you identify these protein-protein interactions, and maybe tell that there is an interaction that is happening and understand the characterize the interaction, map the binding site, map the important interacting residues in the two different or whatever the proteins actually are. So there are different identification methods. So this is a very important portion. Like there can it you can do it like in vivo, in vitro, or in silico nowadays. Like after the computational methods or MD and AlphaFold, then there are certain ML like machine learning AI tools that have been generated that can also predict certain kinds of things. But you need to after all do that experimentally to validate it, because still computational methods are a predictive method, but experimental methods have a more better value. So like for in vivo experimentations, you can do like the yeast to hybrid system, or even the yeast reverse to hybrid system, using split ubiquitin system, split fluorescent proteins like the yellow fluorescent protein, GFP, green fluorescent protein, RFP, red fluorescent proteins, and all. And the split galactosidase system or the lactamase system, even the split luciferase also works very well for these cases. Now, the other method is the co-immunoprecipitation using tagged fusion proteins, X-ray diffraction, biocore, phage display. There are other tools like you can do using NMR and all. For computational in silico methods, there are certain tools like the Bind, Dip, Mint, Intact, and nowadays you have AlphaFold that can also predict the binding of these structures, but what you need is the characteristic features, the exact mechanism, and all. So the methods to investigate the protein-protein interactions is the immunoprecipitation, then protein microarrays, analytical ultracentrifugation, fluorescence spectroscopy, light scattering, RETs, then SPR, protein fragments, complementation assay, calorimetry, ITC. Like other two prominent methods includes the affinity purification, then Yeast-2 hybrid screening. So these are the different methods you can use to investigate the protein-protein interactions. So the Yeast-2 hybrid system is for testing the physical interactions between the two proteins using the Saccharomyces cerevisiae as a biological model by Fields and Song. So you have this thing includes a bet, a prey, and a reporter gene. So the bet is basically the protein that is fused to the DBD domain, that is the that acts as the bet or the yeast transcription factor like the GAL4. So it's a DNA binding domain that binds to the cognate DNA, and then you have a prey, that's the protein that's fused to the ET or the detection. You need a detection domain, right? So you have the LAGZ reporter or the blue white screening that will actually tell you, okay, so this thing has actually worked out. So you have the activation domain, that's the AD domain. You have the DBD, the DNA binding domain. So these three actually work hand in hand to actually carry out the transcription. So when you have the bait that is attached to the DNA binding domain, you have the prey attached to the activation domain, only then the transcription gets activated and the reporter gene is expressed. So the basic principle is that you have if protein X and protein Y interact, then the DNA binding domain and the activation domain will combine to form a functional transcriptional activator. The transcriptional activator will then proceed to transcribe the reporter genes that is paired with its promoter sequence. So you have the BET plasmid where you have the GAL4 binding domain. So that's the commonly used one. So you have the GAL4 binding domain and you have X, that's one protein, and then that's the N and the C terminus. Followed by you have a GAL4 activation domain and another protein Y. So when it expresses like an entire thing. So when X and Y are interacting, it means that GAL4 binding domain and the activation domain will also interact to form a transcriptional activator. So therefore you will see the expression of the downstream genes or the reporter genes. So here the selection genes encoding for the plasmids like histidine, leucine, and tryptophan. So if you are using, for example, a plasmid or a bacterial strain that by itself cannot produce like histidine, or it's an auxotrope of histidine and auxotrope of leucine and tryptophan, then you can easily screen for the plasmids that where the X and Y are actually interacting. But for that you need to first knock down histidine, leucine, and the tryptophan genes, or what you need to do is use an auxotropic strain of bacteria. The other very important technique is the co-immunoprecipitation. So like it's a classic technology used for identification and validation of the different protein-protein interactions. So there are new binding partners, binding affinities, the kinetics of the binding and the function of the target protein can be studied. So So the advantage of this technology is that both the prey and the bet proteins are in the native conformation in the co-immunoprecipitation assay, so you can use them without any kinds of attaching them to certain activator or binding domains. So the interaction between the bet and the prey proteins happens in vivo with little or no external influence. So the limitation of this technology is that if there is a low affinity or transient interaction happening between the proteins, then it might become quite tricky to be detected, because it's immunoprecipitation. So you have to precipitate the protein and then carry out Western blot or maybe an ELISA type of experiments to actually detect them. Now, the other so this is a type of a chromatography, but it will also give you an idea about the protein-protein interaction. But this is moreover of a standard purification technique more than a identification technique. But if it works like for in both the cases like a purification and um, a detection, then it's absolutely fine. So it's a method for separating a mixture of proteins and nucleic acids by specific interactions with those molecules with a component known as ligand. So affinity, it means that there is an interaction that is happening between two particular things. So if you're using a column where you have immobilized it with a particular protein like GST, glutathione transferase, or you have the Staphylococcus protein A, MBP, maltose binding proteins, then these are certain proteins to which have very high affinity for its counterparts or different ligands. So these ligands are basically proteins in today's chapter, but it can be like small molecules or peptides, and then certain carbohydrates, lipids, it can do, depending on what type of a stationary phase or what is the material that the column is actually made up of. Now, if a mixture of proteins is passed through that column, one of the proteins bind to the ligand on the basis of the specificity and high affinity, and they fit like a lock and key model, and the other proteins in the solution just wash through the column, because they will not be able to the bind to that particular ligand. Now the applications is the identify novel protein-protein interactions which might not be. So for other types of interactions, the characterization is easy when you know the two proteins. So if you are aware of which two proteins interact, it's easy to know. But when you are not aware of whether which are the two proteins or which a combination of proteins are actually interacting, then you can actually identify the novel protein-protein interactions using this technique. You can characterize the interactions that are already known to occur, like the protein domains, conditions of the interactions, which is also a very critical factor in this. And then you can manipulate the protein-protein interactions in an attempt to understand its biological relevance. Now, to know how mutation affects a protein's interaction with other proteins, this technique also works absolutely fine. Now, there are certain protein interaction database which you can search through. So, there is a collected information in the specialized biological databases like the primary database, the meta database, and the prediction databases. So the primary database will give you information about the published protein-protein interactions like the biomolecular interaction network, the bind, biogrind, HBRD, where people have done small-scale, large-scale experimental methods to show the interactions. Metadatabase will give you a primary and an original data. So it's an APID, the microbial, PINA, GPS prot. This will give you the the original data, which has been revalidated after being published. And the prediction databases, so it's predicted using several techniques like there are certain databases like the pips, where you have the interaction prediction database, the string, then unified human interactive things, so UniHI. So these are the ones which you can search through to uh, understand or study about some unknown or novel protein-protein interactions. Now, so these protein-protein interaction methodologies have been developed in yeast methods and are not sometimes suitable for different plant systems. Then it becomes quite challenging. Proteomic approaches are still very challenging, and the International Plant Proteomics Organization takes a global initiative to develop and improve the connections between the plant proteomic researchers and the different related fields. Okay. So, any questions up to here? I'll give you like 5 minutes to maybe, uh, if you have any doubts or questions, you can ask me. Because after this, we will start with the question and answers. So, like, you can ask me in the chat box. And also, like, there will be an extra class in the next week, so the date will be informed to you by the NPTEL team. So it's a extra session where you can, where I will be like talking about a few things that I have missed or I wanted to just share with you. So there will be an extra class of like from 6 o'clock only, but the date will be informed to you all. And there you can actually also have the doubt clearing session. It will be more of a doubt clearing session, but there will be a few portions which I will try to cover up such that we do not miss on any part before the exam. Maybe in the next week we will have one of that, because anyways the next week is the last week where we can actually have an interaction session. Okay then. So if you do not have any questions, we can start with the practice problems then. So I would want you to give me answers. After that, we can discuss um, them. That's okay. Okay. So the first question, you can answer me in the chat box. So which of the following is or are correct about the protein-protein interfaces? So the protein-protein interface is 50% polar, is around 50% polar. Polar residues in the interface are usually charged. The protein-protein interface is richer in aromatic residues. Protein-protein interfaces exhibit both shape and electrostatic complementarity. Which of the following points are correct? So are you guys trying? No? Okay, let me see. Okay, so the first point is the protein-protein interface is around 50% polar. That is correct because there are certain hydrogen bonding interactions that happen. So these interactions are very much prominent. Okay, so yeah, point one is right. You have like the polar residues in the interface are usually charged. This is not entirely true because we know that electrostatic interactions are a part of the interaction, but for that to be charged cannot be an important criteria. So we cannot go by point number two. So the other one is richer in aromatic residues. That is also correct because of the hydrophobic interactions that play an important role, and protein-protein interface exhibit both shape and electrostatic complementarity. That is also true because either it has to be a lock-in key model or an induced fit model, so the shape has to be complementarity as well as similar charges cannot be there in both the proteins or in the interface region. So 1, 3 and 4 are correct, so that is C. So C is the correct option over here. Next one, it says monoclonal antibodies act as modulator for targeting a large protein-protein interface. They are used in screening of recombinants, treatment of cancer as diagnostic kits. Which is correct? 1, 2, 3, 1, 2, 2, 3, 1, 3. So monoclonal antibodies can be used for all the three: treatment, diagnostic kits, and even for if you want to screen the recombinants, you can use them. Therefore, it's a very attractive target in protein-protein interface. They're even big. But one major drawback that monoclonal antibodies face is its cell permeability. So it cannot go inside the cell because it's so large, it's a big protein. So it finds difficult to enter the cell, and it's the big drawback in certain drugs, but it can play a diagnostic kits in if it's an extracellular kind of an interaction. Which among the following is or are correct about the SRC homology domain? You can like it is one of the two small protein binding domains. It's a non-receptor tyrosine kinase domain. It includes an SH2 domain, SH3 domain, and a tryptophan kinase domain. And activated SRC regulates the activity of matrix metalloproteinase 9. So which of the following are correct? You guys should try to answer. Otherwise, it becomes very difficult. I understand you might not know all of them, but yeah, you can actually try. It will actually boost up your confidence in one way. It's not a big deal if you are telling it wrong or something. So SRC homology domain, it is one of the two small protein binding domains found in the SRC oncoprotein. That is right. SRC is a non-receptor tyrosine kinase domain protein. That is also correct. It includes an SH2 domain, 3 domain, and a tryptophan kinase domain. No, that is not correct. Tryptophan kinase domain is not present. And activated SRC regulates the activity of matrix metalloproteinase 9. That is correct. So 1, 2, and 4 are correct. That is option C. Now, which of the following is or are not correct about phosphotyrosine binding domains, the PTB domains? It interacts with sequences containing the phosphotyrosine group. The insulin receptor substrate contains these domains, situated at the C terminus, and it has a compact 7-stranded beta sandwich structure capped by N-terminal helix. So which of the following is or are not correct about the PTB domain? So the PTB domain interacts with sequences containing the phosphotyrosine group. That is correct. That's an ideal activity of a PTB domain. Now, the insulin receptor substrate contains these domains, and we know that because the IRS, or the insulin receptor substrate, is a very well known example of the PTB domains. Now, it is situated at the C terminus is not correct because it's situated at the N terminus of the particular protein, and it has a compact 7-stranded beta sandwich structure. That is also not correct because it gives more of an information about the SH2 domains, not a PTB domain. So 3 and 4 are incorrect. That is option B. Okay. So you have to identify these different domains. So whether it's a beta sandwich structure or a double zinc finger structure or a double hairpin structure. So you have to identify these different domains. So the first one is a, you can see from there, it's a beta sandwich structure. Rich in beta strands. Okay, so it's a beta sandwich structure, and the last one is also very simple because it's a double zinc finger structure. Okay, so there is a zinc, you can see there are two zinc ions present, and the middle one is a double hairpin structure. You can see you use hairpins in your when you tie up hairs or do like that. So it's a double hairpin. So there are two hairpins present. So it's a double hairpin. So one is beta sandwich, two is double hairpin, three is double zinc finger structure. That is option C. Now, peptidomimetics are used for. So what do we use peptidomimetics for? identification of tight binding potential peptide sequence, modification of the peptide to enhance stability, uptake, and delivery, development of drug-like compounds from existing peptides. So what is basically peptidomimetics? So peptidomimetics is the study of peptides. Now when you are studying peptides, what do? We identify the different characteristics of the peptides and its binding to a different protein or different sequence, so you can get an idea about the kinetic, thermodynamic parameters. So that includes the binding potential as well. So then you can, what you do is you engineer that particular peptide by modifying the peptide such that the stability, the uptake, and the delivery can be improved. And then you have the development of drug-like compounds from the existing peptides. So because the peptides we know that are fantastic therapeutics, so be it linear or cyclic. So 1, 2, and 3 all are correct. So option C. Stapled peptides bind to estrogen receptor using a hydrocarbon link to stabilize them in the form of a or an beta sheet, random coil, turn, or an alpha helix. So how does the staple peptides bind to the estrogen receptor? So the correct one is alpha helix. That is option D. So alpha helix, it forms the hydrocarbon link is through an alpha helix, and then this particular peptide binds to the estrogen receptor. Now, which among the following is or are incorrect about the ACE inhibitors? That's an interesting question. So we have to find the incorrect statements about the ACE inhibitors. So what are ACE inhibitors? So ACE is a angiotensin converting enzyme, that is a metallopeptidase. Mostly it's a dipeptidyl carboxypeptidase known as kininase 2. It cleaves the dipeptides from the C terminus of the substrate such as angiotensin 1 and bradykinin. While it has a range of substrates, it exhibits specificity in its enzymatic action. So it's a highly specific oxypeptidase. Option 3 is correct. However, option 1 is not correct because angiotensin inhibitors or the ACE inhibitors block the enzyme ACE, which is responsible for converting angiotensin 1 to angiotensin 2. Therefore, ACE inhibitors reduce the production of angiotensin, that they do not produce it. And angiotensin 2 does not cause vasoconstriction and increase the blood pressure, but the action is of the ACE inhibitors is to counteract this effect by reducing the angiotensin 2 levels. So that's would be a more correct way of telling it. So option 1 is incorrect. Option 2 is also incorrect because the correct pathway in the rash, that is the renin angiotensin aldosterone system, is that the angiotensinogen is converted to angiotensin 1 by the enzyme renin. Now then angiotensin 1 is converted to angiotensin 2 by AC, that's the angiotensin converting enzyme. But what we see over here is that they have converted the rules of 1 and 2. So 1 and 2 are both incorrect. That is option B. Now choose the correct statements about the protein corona formed on nanoparticles. So soft corona is formed by nanoparticle bound protein-protein interactions. It's formed by protein solvent interactions. The hard corona is formed by nanoparticle protein interactions, or hard corona is formed by covalent bonding with nanoparticles only. So the correct one would be 1 and 3. That is, soft corona is formed by nanoparticle bound protein-protein interactions, and hard corona is formed by nanoparticle protein interactions. The next one says SRs. So next one is a match the following. So where you have SH3, SDM, LIM, and PDZ. So you have to match them using the following ones. So let's do it then. SRC homology 3. That is, yeah, SH3 binds to proline rich sequence and target proteins. I was saying it. So one is Y. So it can be like C or D. Okay. SDM. So SDM, it binds to non-SAM domain containing proteins, that is also RNA. LIM. LIM is, it binds to helix loop helix motifs. And the PDZ, that binds to that recognizes the serine threonine XV motif, where X can be any of the 20 naturally occurring amino acids. Okay, great. So any questions? So that's the last question from my side. Any questions you have? No questions then? Okay, thank you very much for joining. We will again meet. Let's see if we have an extra class. Maybe we will have it before Friday sometime. The NPTEL team will let you know about the timings and all. So then we will meet. Otherwise, we have as usual a regular class from Friday 6 p.m. Okay. Okay then. Goodbye.
Thank
Thank you.
you. Thank you, sir.